Thank you. It's great to be back at OIS and to share some exciting updates about Valitor. So Valitor's mission was originally to develop long-acting biologic anti-VEGF treatments for wet AMD. And since we've started this mission, I think we've seen that intravitreal injections have become very safe, very routine, so much so that there have been new medicines that have come to the market that take advantage of this route, and there continue to be new medicines under development that take advantage of this route. And while the route is useful, it also creates a high healthcare burden, not only for patients, but also for physicians, and at a high treatment cost. So our mission's really expanded in the last couple of years to be bringing this strategy for durability and back-of-the-eye diseases, not just for wet AMD, but for other indications, and with the goal of just 1 to 2 treatments per year. The technology that we've developed for achieving this mission we call multivalent polymer conjugation. We can take multiple copies of a variety of types of drug moieties and conjugate them to long-chain biopolymers to get these tethered clusters of covalently bound drug around a biopolymer. And I want to be clear here, this is not a slow-release vehicle or a depot. We want the drug to remain bound to the biopolymer as it's being cleared out through the eye and eventually eliminated out of the ocular tissues. And in this way, we can use the payload to define the specificity of the drug and the biopolymer to determine the PK properties. And I'm going to drill down on this a little bit because the idea of using biopolymers to improve durability isn't new. Conventionally, you would use a single drug and conjugate 1 or more biopolymers to it in order to get the size you need for the durability the indication requires. But there's a limit to this. As you add more biopolymer, you increase the risk that the biopolymer is going to interfere with the drug's activity, and you're also creating complex branch structures that inhibit the ability of that drug to get into tissues like the retina. Valitor turns this method around. So we take a single biopolymer, and we can make it as long as we need to in order to get the durability we want. And as long as we're also increasing the copy number of the drugs along that backbone, we don't have to worry about drug interference. And in fact, we can actually coordinate multivalent interactions between the drug and its target that in some cases can increase the potency of the drug. We also know these linear biopolymers are able to penetrate into tissues like the retina. So this is the core of the technology that we use for all of our intravitreal medicines. And our lead product is still a long-acting anti-VEGF for wet AMD. Our goal here is to combine the gold standard of anti-VEGF biologics with our orthogonal strategy for improving durability in the eye. And this is different than anything that's previously been investigated. For the biopolymer, we use hyaluronic acid. We like hyaluronic acid because it's endogenous and biocompatible to the back of the eye. And it takes on these huge hydrated structures that really enable us to maximize the entanglements and interactions with the matrix in the vitreous humor. And so it's the size of this biopolymer that really determines the rate it's cleared out of the back of the eye. The anti-VEGF we use is a single domain antibody. This is the smallest, most stable unit we can use to define the specificity of the anti-VEGF. And again, we preserve the potency by maintaining a high copy number on the backbone. So today, I'm excited to share 2 new preclinical data points that we have that demonstrate the benefits of VLTR-559. The first is a PD study. This is the laser-induced choroidal neovascularization model in transgenic mice. So these mice express human VEGF, and it enables us to do a direct head-to-head comparison between 559 and Eylea. And you can see from this plot the ability of 559 to inhibit that neovascular response driven by the VEGF is equivalent for both molecules. This is consistent with all of our cell bioassay data demonstrating that VLTR-559 has equivalent potency to all the other drugs that are on the market. This next study demonstrates how we differentiate ourselves in the market, and that's on durability. And this data is less than a week old, so you guys are the first ones to see it. It affirmed that we're seeing a half-life of 12 to 15 days in this model, which is at least 3 times longer than the drugs that are currently on the market. And we can project forward to predict how this will impact patients with wet AMD using these clinical efficacy models. And in this model, we've looked at all the past registration studies in order to define thresholds at which we can predict how many patients are still treatment-free as the drug is being eliminated from the eye. And based on these models, we expect that most, if not all, patients will receive the benefits of 6 months treat and release durability, and some may be able to go beyond that. This will be a huge improvement over current drugs on the market, like Eylea high dose. So VLTR-559 is our lead product. We're excited about what we're going to be able to do for patient care with patients who have wet AMD. But we've also designed this platform to be interchangeable for rapid expansion of our pipeline. And so as an example, we're already working with 2 pharma companies who have 2 different assets that need differentiation in their respective markets. We've demonstrated conjugation feasibility, and we're continuing to develop these towards clinical development. Additionally, we have an in-house second asset that follows a clinically validated mechanism and again enables us to take advantage of the durability and bring a longer acting version of a medicine that we know works in patients. So from our perspective, there's no reason to stop there. This is a crank we can keep turning. As new clinical mechanisms become validated, we can follow the established development pipelines and deliver long-acting versions of those drugs to the established patient populations who will need them. So with that, and in summary, I'd like to thank our investors. We've appreciated all their support to date. We're excited about the potential for our lead product, VLTR-559, for wet AMD. We also are really looking forward to the way we're able to expand our pipeline, both with internal and partnered assets. So if this sounds interesting to anybody, please come find me during one of the networking breaks. We'd really always look for the opportunities to find new industry collaborators, and I want to thank all of you for your attention. Thank you.